Gram staining method for microbial differentiation. Gram staining is the common, important, and most used differential staining techniques in microbiology, which was introduced by Danish bacteriologist Hans Christian Gram in 1884. This test differentiates the bacteria into gram-positive and gram-negative bacteria, which helps in the classification and differentiations of microorganisms. When the bacteria is stained with primary stain crystal violet and fixed by the mordant, some of the bacteria are able to retain the primary stain and some are decolorized by alcohol. The cell walls of gram-positive bacteria have a thick layer of protein-sugar complexes called peptidoglycan and lipid content is low. Decolorizing the cell causes this thick cell wall to dehydrate and shrink, which closes the pores in the cell wall and prevents the stain from exiting the cell. So the ethanol, in case of gram-negative bacteria, cell wall also takes up the CV iodine complex, but due to the thin layer of peptidoglycan and thick outer layer which is formed of lipids, CV iodine complex gets washed off. When they are exposed to alcohol, decolorizer dissolves the lipids in the cell walls, which allows the crystal violet iodine complex to leach out of the cells. Then when again stained with safranin, they take the stain and appears red in color. Cross-section of cell wall gram-positive and negative bacteria. Reagents used in gram staining. Crystal violet, the primary stain. Iodine, the mordant. A decolorizer made of acetone and alcohol, 95%. Safranin, the counterstain. Procedure. First step, take a clean, grease-free slide. Second, prepare the smear of suspension on the clean slide with a loop full of sample. Third, air dry and heat fix. Fourth, crystal violet was poured and kept for about 30 seconds to 1 minute and rinse with water. Fifth, flood the grams iodine for 1 minute and wash with water. Then, wash with 95% alcohol or acetone for about 10 to 20 seconds and rinse with water. Seventh step, add safranin for about 1 minute and wash with water. Eighth, air dry, blot dry and observe under microscope. Interpretation. Gram positive, blue or purple color. Gram negative, red or pink color. Examples of gram-positive bacteria: Actinomyces, Bacillus, Clostridium, Corynebacterium, Enterococcus, Gardnerella, Lactobacillus, Listeria, Mycoplasma, Nocardia, Staphylococcus, Streptococcus, Streptomyces, etc. Thank you guys, see you again.